replication, and that would be from DNA whole 3. If we were to make a whole exact copy. But now we're moving from DNA, and we're taking this original DNA strand and making an RNA. This is transcription. It's the first step, is DNA to RNA, and that's done by RNA polymerase. And then now we're gonna go here and synthesize our amino acids. So this is translation, and this is RNA to amino acids, and that's done by what enzyme?
RNA polymerase. No, wait a second. Ribosomes.
Ribosomes. It's our only one that doesn't sound like ACE. That's a ribosome. A ribosome really is an enzyme. It's kind of a coordinator enzyme that pulls in the tRNAs. Okay, so translation, RNA to amino acid, that's ribosomes. Those are our 70S ribosomes. That's important, don't forget that. Okay, so here let's do UAG together. What is that? Stop. Oh, that doesn't work. Okay, just pretend it doesn't say stop. This is an amino acid. Okay, then what's our next one?
Serine?
Yep, serine. What would be our next one? UAC.
Oh, sorry, that's probably not serine then. What was the 3?
So UAG, GCG, UAC.
Tyrosine.
Oh, the second one is tyrosine?
Yeah, my bad.
That's okay. And then what's the last one? UAC.
No, the last one is tyrosine, sorry.
GCG is alanine.
Yeah.
GCG is what?
Alanine.
Alanine. Okay, so this should be backwards. The stop codon should be at the end. So that's how you assemble amino acids together that form a protein. I didn't ask you about this, but it will come up again, is that there is COOHR. So an amino acid, all these amino acids have a different R group, and that different R group is what makes them different. So alanine is different than tyrosine, is different than valine, is different than leucine, because it has a different R group attached to it. That's what an amino acid is. So, for example, let me Google it, because I can't remember. How many amino acids for ATP synthase? 480, that's not right. This is why Google sucks, but it's not 480 amino acids. It's around 5,000 amino acids or something like that. Polypeptide chain, maybe that's it. Bacteria? Is that why the number is different? 120 to 140 amino acids? Apparently. So I would click on the link. If I ever wasn't sure, I'd click on the link and figure out where that came from. So Googling things is always scary, but okay, so those amino acids that come together form things, and because they form things, that's what forms our entire body and what makes everything that we are. So just because we have a phospholipid bilayer around our cells, some amino acid group of protein somewhere assembled that and put that together. So we have proteins and enzymes that put everything together, that cut things apart and put things together, and that's what essentially that we're made out of. Okay, does anybody have any other questions? Yeah.
Can you clarify the difference between a genome and a genotype?
Yeah, so the genome is when you say your genome, you're saying like your entire grandma's book, and when you say a genotype, you're saying ATGC order. So you're talking about what's in it versus the whole thing. You're kind of basically saying the same thing, but my genotype would be AATC, where yours would be AATG. So because we have the single nucleotide polymorphism, so that's our genotype difference, but my genome is like something you'd call the whole thing all together.
Okay.
Yeah. Okay, yeah.
Can you clarify, I'm not sure if you touched on it, but what it means for for something to be competent to be able to take up DNA?
Oh yes, awesome. Yeah, so we're gonna do that in the lab. So when there's horizontal gene transfer, remember there's conjugation and transformation are two main ones. So conjugation happens with the sex pilus, and the plasmid or bacterial genome will be horizontally transferred from one bacterium to another. And then what will happen is extracellular DNA from some other bacteria that died can just be laying around here, but they can't get in unless this bacteria has some special method that it can get in there, or if there's a lot of heat, like in fever. So then it will make the outer membrane of peptidoglycan and cytoplasmic membrane a little bit more loose, where this bacterial culture, the bacterial extrachromosomal bacteria or extracellular bacteria will get inside. So that's what it's being made competent, or it is competent. It's like it's able to take up extracellular DNA. Yeah, so we're gonna induce competency by heat shocking. So we're gonna increase the bacteria, the temperature of the fluid that the bacteria is in, and then we're going to put it on ice, and then we're gonna heat it up again to make these competent to get the DNA inside of there. That's what we're gonna do in the lab.
What was the third one called?
The third one are transposons, and that's just gene switching. So when the plasmid can get into the chromosomal DNA, or the chromosomal DNA can get to the plasmid, it's just gene jumping.
Okay, and that's different from high frequency, because high frequency is only from the plasmid into the chromosomes?
High frequency is, when there's plasmid DNA in a chromosome.
Okay.
Yeah. I'm sorry, did you have a question? Yeah.
Is RNA formed in the cytoplasm?
Yes, RNA is formed and just moves around the cytoplasm in bacteria, which is not true of human cells. But great question. Okay. Does anybody else have any more questions? How do you guys feel about gene regulation? Well, I can trip operon, you want to go over it one more time? Or would you rather go over mutations? Would you rather go over, pick one? Mutations, okay. So let's write out our mutations. So these ones I'm not gonna write out because it's self-explanatory. I just wanted to throw it out there. So let's talk about our names of our point mutations. So out of the names of our point mutations, we have, here, let's put them all together. Frame shift, silent, missense, nonsense. So if you had a frame shift mutation, so let's say we have ATCCCA. And then from here, we go to the RNA. Okay, so here's our RNA strand. So if there was a frame shift, there's gonna be one left over. Now it's gonna be UA, let's say this is a G. Now it's gonna be UAC and GU. So see how it changed this amino acid sequence, and now we've got a leftover weird one that's gonna shift them all down. So that's a frame shift. A frame shift is when you move the codon.
So is the pink still the mRNA?
Down. Yeah, this is the new strand of RNA that's broken.
Okay.
Yeah, so this new strand of RNA, so see here, every single one is gonna be shifted from here on out because you inserted something. So it's just like, you know, if you put all the beads on a string in the right color, like red, green, blue, red, green, blue, red, green, blue. If you put a red in there, it wouldn't be red, green, blue anymore. It would be like blue, green, red, blue, green, red. So it would throw off your 3 pairs. Okay, so that's frame shift. What do we have? Silent mutation. So a silent mutation would be because there is redundancy in, so we're on number 70. Because there's redundancy in the codon chart, which means that CCU and CCC can code for proline. So if there's a mistake in this last base pair, then there is no change in the amino acid. So that's a silent mutation. A silent mutation is when proline is switched for proline, same amino acid, but because of that redundancy in the codon chart. Does that make sense?
And that would also at the same time be a point mutation or whatever the other.
Yeah, point just means one amino acid.
Right, so if a point mutation caused a silent mutation, that mutation you can still refer to as both.
Yes, yes, yeah. So, yeah, these are all point mutations because they're just one amino acid, yeah. Okay, then a missense mutation is where these first 2 amino acids would be altered, giving it a whole new amino acid. So either one here or one here, giving it a different amino acid. So the amino acid is now valine instead of leucine. So we don't know how that affects the protein. It depends. So it could make the protein more efficient and stronger, or it could break the whole protein altogether. So the problem is if you have a mutation in the DNA, it's gonna put valine there from now on, because every single base pair, if the mistake happened here, the mistake is gonna be here and here now. And here, 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 and here. So now that mutation is heritable, it's gonna be passed down for every replication from here on out. So that would be a valine in for a leucine. That's a missense mutation where the amino acid has changed. Yeah? Okay, and then the last one is a premature stop codon. That's a nonsense mutation. When it puts in for a stop codon accidentally instead of any amino acid that it was supposed to. So if it accidentally puts in here a UGA or not UGG, that's tryptophan, UGA, UAA, UAG, then it'll prematurely stop or truncate the protein. So now the protein will probably malfunction. Okay. Okay, so let's go ahead and put everything away. Oh, sorry.
In silent mutation, the amino acid sequence doesn't change, right?
In a silent mutation, the amino acid does not change. Great question.